Hi, welcome back. This is Coach Heichel. I'm bringing you Unit 8 over viruses. So this is Unit 8.1. There'll be 3 more units to watch within this unit. There'll be one over bacteria, funguses, and protista. So look for those to come here pretty quickly as well. So let's get started with viruses. First of all, we're going to start with how to name a virus. When you name a virus, you name it after the disease that it causes or the organs that it specifically attacks. So when you look at the poliovirus is called poliovirus because it causes polio. Smallpox virus, again, is called smallpox because it's what it causes. It causes a smallpox virus. If a virus infects a bacterium, it's called a bacteriophage, or sometimes it's called just a phage for short. So bacteriophage, again, affects a bacteria. Viral metabolism. Viruses, a lot of people think they're living. Most people think they're not living, though, however, because viruses cannot grow. They cannot reproduce, and they don't even develop unless they're in a host cell. They have what's called a parasitic relationship with their host. And if you think about a parasitic relationship between humans and viruses, viruses use our cells, all of our energy, all of our food, everything our cells do to reproduce them, and in turn, they kill our cells, which eventually could lead to our death as well. So it's a very parasitic relationship. So, some more of what viruses can't do. Again, viruses don't have any metabolism. They cannot reproduce on their own. They don't move. They have no respiration. They don't even eat any food. And they don't have any cellular structures. So most scientists will agree that viruses are not living. Again, they have to be in a host cell for everything to be done for them. So the basic 4 characteristics of a virus are viruses are made up of what's called nucleic acids, which are going to be your DNA or RNAs. These are going to be enclosed in what's called a protein coat or a capsid. They're smaller than the smallest bacterium, so we can't see them in our labs because our microscopes don't have enough resolution or enough magnification to do this. They're non-living particles, and their shape determines what cells they're actually going to attack. And below here is a picture of the 3 most common ones we're going to talk about. We're going to talk about bacteriophages, we're going to talk about the tobacco mosaic virus, and we're going to talk about HIV or human immunodeficiency virus. So, continuing on with viral structures. So we have a capsid. These capsids are going to surround the nucleic acids. So I'm going to circle the capsid here. It's going to be this big blue pill-looking thing in the middle of the viral structure. Inside of this is going to be the DNA and RNA. So the capsid is the big blue thing. The little pink, purple squiggly lines going through it are going to be the DNA or RNA, depending on what type of virus it is. And here on the outside with these spikes, the green area, this part right here, is going to be your nuclear envelope. Now the reason these spikes are attached to it is because these spikes are used to attach to larger things, kind of like humans. They're such a large virus, they gotta have ways to attach to human cells. So these are some basic shapes. Again, I told you that we're gonna talk about bacteriophages. Well, bacteriophages are just big, tall, walking water towers. That's what they look like. Okay, they have these tail fibers, they have tail sheets, head that contains the DNA. This is what one looks under a microscope. Okay, tobacco mosaic virus, here's a cartoon drawing of it. Here's what it actually looks like under a microscope. So we're going to talk about tobacco mosaic virus here in a second. And if you want some extra credit, you can draw and label me a bacteriophage, turn it into me, and I'll give you some extra credit points. So let's continue forward. Again, viruses are specific to what they're going to infect. They only infect specific host cells. Their shape of their capsids is going to determine what type of cells they're actually going to attack. So for example, HIV is a human virus. You'll never see a dog or a cat have HIV because it is a human virus only. Some viruses, however, do cross over, kind of like the rabies virus. Humans can get rabies from animals, and so on and so forth. But HIV, you'll never see it in dogs and cats. So viruses have to replicate somehow, and this is how they do it. They do it through a step called the Lactic Cycle. Okay? The first step is going to be the virus is going to have to attach to the host cell with matching receptors. So it's floating around inside your body. It's going to keep floating until it sees something that matches what it has. Once it does that, it's going to release its genetic or viral material either through an injection, kind of like a shot, or through exocytosis, which is pinching it out of the cell. Okay? Once it does that, our cells, or the host cells, are going to copy that viral material. It's going to remain in our cells. It's going to mature. It's going to replicate. It's going to become a full virus. And then once there's too many of them inside the cell is going to go through what's called lysis, or it's going to burst, and it's going to release these viral cells out to the rest of your body. So the lactic cycle is rapid replication and assembly, followed by what's called lysis or bursting. Okay, it's going to release these viruses out into the world so they can go infect more cells. And on the next slide, we're going to see an animation of the lactic cycle. So it's important to know the steps of the lactic cycle. So here is a short animation of what actually happens. So this guy right here is just going to be our bacteriophage floating around. It sees these two receptors, great, it can attach to them, so it does. Then it's going to release its viral or genetic material into the bacteria. Okay? We know this is a bacteria because it doesn't have a nucleus, it's a prokaryote. So here's the viral DNA now. It's going to use this bacteria's structures to make more bacteriophages. It's going to replicate, it's going to mature, it's going to assemble them. Once there's too many of them to the bacteria support, it's going to release them through a process called lysis, and they're going to float around in space until they find more cells to infect. So that's the lactic cycle. That is the lactic cycle. So the lysogenic cycle is very identical to the lactic cycle with some minor tweaks. Okay, so the lactic cycle is how it replicates. The lysogenic cycle is how it replicates, but this is what happens whenever the viruses go into harsh conditions. The conditions aren't favorable for them to reproduce, so they don't reproduce at this exact time. Think about mononucleosis and how it can be latent in your body for many years before you even know you have it. That's because the virus goes into what's called the lysogenic cycle, remains dormant until an environmental factor triggers it to start the replication and the maturation process. So it's going to have the attachment, it's going to have the infection, but instead of the instantaneous rapid replication and maturation, it's going to insert its DNA into the host cell's DNA. It's going to remain dormant until something happens, and then these prophages are going to come back at a later time, and they're going to replicate, they're going to mature, and they're going to release, just like in the lactic cycle. But lysogenic happens when there's harsh or non-favorable conditions. So just remember that. That's the main difference, harsh or non-favorable conditions. So this is just a basic comparison. So the lactic cycle, everything goes to the lactic cycle, okay? Whether it's in the lysogenic or the lactic. The big difference is the lysogenic is it forms the provirus or the prophage, and it's going to remain dormant until it needs to divide later. So here's a basic animation of what happens in the lysogenic cycle. Again, you have this bacteriophage floating around in space, sees these receptors, it attaches. It's going to inject its viral DNA. Once it does this, it's going to form what's called a prophage. It's going to form it right around this area. This DNA is going to move over here. It's going to snip out this plasmid, and it's going to form what's called a prophase. Once it does that, it's going to remain dormant until something happens. Okay, something's going to spontaneously make it start to grow. Once it does that, it's going to re-enter the lactic cycle. It's going to start growing again, making viruses, let them fully mature, fully replicate. There's going to be too many of them for the cell to support, and it's going to go through lysis again. It's going to rupture. These viral structures are going to go infect more cells. So that's just a basic overview of the lactic and the lysogenic cycle. It's going to be important to know the steps. Here's an example of a few lysogenic viral diseases. There's herpes simplex 1 and herpes simplex 2. Herpes simplex 1 is just simply cold sores on your mouth, while opposed to herpes simplex 2, which causes genital warts. So these genital warts come and go in phases. They're not permanently there, as in they're not permanently seen on the skin. You permanently have herpes once you get it, but you don't always have the outbreaks. You don't always have cold sores, or you don't always have warts on your genitals. Hepatitis B is also a virus. It's caused by hepatitis B. Chickenpox is a virus. It is usually lysogenic, but some of the time it can become lytic, which causes shingles, which is a lot more dangerous for you to have than chickenpox. Most of y'all don't know what chickenpox is because there's a vaccine for it now, and we're going to discuss what vaccines are in a few more slides. So just a basic overview of a few viruses. We got HIV, which is human immunodeficiency virus. This causes a condition called AIDS. So we can treat the virus, but we can't treat the disease. That's what makes HIV and AIDS so deadly worldwide, is because most people don't know they have HIV until it becomes full-blown AIDS. So what HIV does is it destroys what's called T cells. Your T cells are part of your immune system that help your white blood cells fight off infections. Well, it destroys these cells. Your body can't fight off infections. Your immune system becomes deficient, and a common cold can kill you, for example. So this happens when the T cell count drops to a particular level. Then the person is said to have the full-blown disease, not just the virus. This is just a microscopic picture of what the virus looks like. Again, what we're seeing, we're looking at these little circles. These circles right here are going to be the capsomeres or the capsids. That's the part that you're going to see most of the time. HIV is a retrovirus. We know it's a retrovirus because it has RNA instead of DNA. If you remember from the first unit, when we talked about DNA and RNA, RNA is single-stranded as opposed to DNA, which is double-stranded. So once the HIV infects its RNA into the host cell, it has to use an enzyme called reverse transcriptase to make a DNA complement of the RNA. So it's going to be completely opposite of how DNA replicates in the nucleus, makes an RNA form, then leaves and goes to the ribosome. Okay? It's going to have the RNA, and that's all it's going to have to work with, so it's going to make a complementary strand of DNA from the RNA. Plant viruses. The first virus to be identified was a plant virus. It was called tobacco mosaic virus. It causes diseases in tobacco plants. What it does is it causes yellow spots on the tobacco leaves, which makes them undesirable to tobacco farmers. You smoke tobacco, so if it has these yellow spots on it, you probably don't want it. And these plant viruses enter the plant either through the wounds. Think about when a leaf gets nicked, or you rip the bark off of a tree or something. That's actually hurting the plant. Not necessarily pain-wise, but it's actually giving it a wound where it can catch a virus. And also insects can spread these from plant to plant through the pollen. So that takes us on to smallpox. So in 1796, a man named Edward Jenner developed a smallpox vaccine using a milder cowpox virus. He studied these milkmaids who got paid to milk cows. He noticed they had these little lesions or these little pox-looking things on their hands. So what he said, okay, that looks a lot like smallpox. So let's take some blood from these little pox and let's analyze them. So what he did is he looked at them, he compared them back and forth. And he said, since they look alike, I think I'm going to inject the cowpox virus into these people with smallpox. Well, what he was doing, he was using a weaker form of smallpox. And he actually came up with a vaccine to it, and he actually used it on his kids. And we get the word vaccine from cow, because Spanish for cow is vaca. So vaccine comes from the first successful vaccination, or cowpox. Just a little tidbit you can share with your friends. So if a virus is said to be deadly, it's said to be what's called virulent, which means it'll kill you. And smallpox used to be very virulent. It was had a high mortality rate. Most people that got it died of it. But today it's been eradicated due to vaccinations and better healthcare systems. And now we're going to talk about what a vaccine is. There's a little bit of difference between a vaccine and an antibiotic. When you go to the doctor and you have an ear infection, that means you have a bacterial buildup in your ear. So if you have a bacterial infection, you use what's called an antibiotic. Viruses cannot be killed by an antibiotic because they're not living. Anti meaning against, biotic means life. So antibiotics kill life, but since viruses don't have life, you can't kill them with antibiotics. So you gotta treat viruses through what's called a vaccine. And basically what a virus is, think about a mental Rolodex. If you don't know what a Rolodex is, it's like a planner. It has all your phone numbers in it. You flip your A's, your B's, your C's. You have all your contacts and their phone numbers, everything in there ready to go. Or a digital camera with a memory card, okay? So whenever you get a vaccine, your body has a mental picture of what the molecular structure of that virus looks like. It's a weakened virus that they put into your body. If it's weakened, it's most likely not going to make you sick. Most people say it makes them sick, but it's probably mental, or they could have a cold when they get the virus to begin, or when they get the vaccine to begin with. So a virus is just a mental reference that your body recognizes. It says, you've been in me before and you weren't supposed to be here. So the next time I see you, I'm going to fight you off that much faster. I'm not going to have to wait for an immune response because I'm going to already have that memory stored away so that next time I see you, I can treat you. So vaccines are a weakened, less vigorous form of the virus. When you attenuate something, it refers to the process that weakens it, which most of the time it's by heating. And it's less virulent strain of the virus, and it usually does not make you sick. Okay? It is capable of stimulating an immune response and creating immunity. It's a passive immunity because you only need it if you actually need it. Just because you get a flu vaccine doesn't mean you're going to come in contact with the flu virus. But if you do need it, it's there, it's passive, and you have it. It's ready to go in case you need it. So that wraps up 8.1 on viruses. If you have any questions, please feel free to ask. This is Coach Heichel, and have a great day.